Hello everyone. Am I audible?
Yes.
Okay, so I guess students are still joining, so we'll just wait for some time more and then we can start the session. Yeah, tell me. Akashas, you raised a hand. Is there any problem? Okay, so I hope I'm audible, and I think we should start the session, and we'll just let other people come as they join. Okay, is my screen visible to everyone? Just give me a heads up that the screen is visible.
Yes, ma'am.
Okay, so so welcome everyone and good evening. We'll start with the classes. This is actually a discussion session for your coursework of plant developmental biology, which you have been undertaking. Instructor is Professor Sri Ram Yadav from IIT Roorkee, and I am Prashasti Pandey. I am a PMRF scholar, and I'll be taking a discussion sessions throughout this course. And just let me just brief you with the aim for this discussion session. So, these sessions are not to teach you exactly the syllabus, but whatever has been uploaded on the course, you will study that, and there is a assignment which is given at the end of every week, and you are supposed to solve that. So, with these discussion sessions, we are just aiming to give you some sample questions. Uh, they will basically help you in solving the assignments that you are getting at the end of every week. So we'll just solve few sample questions, whatever those questions are, and then we'll try to understand a little concept of why that question, like what that question is, and whatever the answer associated with that. Um, Okay, so I just want to say that I'd really appreciate if you participate in this and try to answer these questions, and so that it'll be an interactive session because it's supposed to be a live session. So just try to answer however much you can, and otherwise, of course, I'll tell you the answer and we can proceed with that. Okay, should I start?
Yes, ma'am. Okay, ma'am.
Okay. So, in this course of plant developmental biology, I'm sure you must have got the syllabus, and I think already the videos and everything has already been uploaded. So we'll just directly start with the questions. So, like one of the sample questions is, which of the following is the male reproductive organ in flowering plant? So we'll just go by this way that the question is on your screen. You can answer me if you know the answer. Just go ahead and answer. If nobody answers, then I'll answer the question and we can then go ahead with the understanding the concept behind it. So which of the following is the male reproductive organ in a flowering plant? Any one of you knows this answer? Anyone?
And question, ma'am.
Sorry, what?
And question.
Correct. So the answer for this question is androecium. Now let's understand the concept of this. There are two terms actually, the reproductive organs in any flowering plant. So as we all know that flowers are only produced after basically they are these are the products of sexual reproduction. Sexual reproduction requires a male reproductive organ and a female reproductive organ. In this case, gynoecium and androecium are the two names for reproductive organ, which is female and male accordingly. Now, to understand this, let's first understand what this word means. What is gynoecium and what is androecium? So, gyne or gyn or gyno is always associated with female. If anyone of you has also studied zoology, Then, like when we go to hospitals, there is a gynecologist. So the gynecologist is supposed to deal with the female reproductive organs. So the gyne word is associated with females. Similarly, andro word is associated with the male reproductive organs. Now, the isham word in both gynoecium and androecium basically represents the arrangement of all the reproductive organs. Correct? So when we are talking about gynoecium, that means we are talking about the arrangements of the female reproductive organs in plants. And when we are talking about androecium, we are talking about male reproductive organs in a flowering plant. So, most of you have, I'm sure, would have studied this or would have seen this. If you have done any practicals to see this organs of a flower, the best flower to study this in real is actually hibiscus flower. So if a hibiscus flower is available anywhere near you, the red color part that is coming out of it, you just remove the petals, and the red color, the stem like part which is there in the middle of the flower, so that is basically the reproductive organ of that flower. If you see that, you'll get the similar structure which you can see here on the screen, which is labeled as female reproductive organ. So I'll just turn on the laser pointer. Okay. So this female reproductive organ. So basically, when you remove the petals of the hibiscus flower, this is what you will see. And at the end of that hibiscus flower, there is a small swollen green color part. And if you cut that with a sharp blade, you will be able to see the ovules. Okay. So basically, ovary and ovules are a part of female reproductive organ. Now, what happens is when you cut this ovary, you will see. So when you take different kinds of flowers, this ovary arrangement varies. Okay, the way those ovules are arranged varies in different kinds of ovaries, on the basis of which the flowers can be differentiated. Now, why ovary is important? Uh, I think I'm sure all of you know that ovary is one of the most important parts of female reproductive organ, because this is the part which contains ovule, and ovule is nothing but the egg. So for fertilization, you require a male cell and a female cell to form a two-celled organism, or which in zoology is called a zygote. So in this case, ovule is always present inside the ovary, from which a male, whatever the male reproductive cell is there, will fuse and then form a basically 2N structure, and then it will develop into fruit. Okay? Fruit is the outcome of the reproduction. So the style and the stigma. Now let's understand what is the role of the stigma and style. For that, let's just first see that what androecium is. So basically, androecium is a very simple structure. It just contains of a filament, and it has anthers attached to it. Now, what anthers have? So in basically in plants, male reproductive cells are called as pollens. Okay, all of us have heard about pollens. So pollens are nothing but male cells. When a pollen cell will fuse with an ovule inside the ovary, that is when the fertilization happens. Now, the problem is with the flower that these two cells are not present in one place. There are various ways by which these two cells can be brought together. So, there are several examples of plants where only the flower is either male or it is female. There are some flowers which have both male and female organs, and some flowers have either male or female organs. Just out of curiosity, does anybody know that why does plant? I mean, how? Why did this evolution happen? That plant chose to put its male and female's organs on different parts and not in one single flower. Can any one of you answer this question? Like, it's not associated with this question, but it's a very interesting thing. Why did the plant choose to place two reproductive organs in different places? In some cases, it's even present on different plants. Is anyone aware of this? Okay. So I just need to know that you don't want to know the answer or you don't know the answer. So I'll proceed accordingly. Okay. So I'll just assume this silence as you don't want to know the answer. So we'll just stick to the syllabus. So basically, what happens is when the pollen sacs, basically, anther contains various pollen sacs which contains pollen cells. So what happens is these pollen cells will actually stick are actually stuck on the anther and they get released in various forms. So if the flower is a bisexual flower and the flower itself has ovary, then the filament is basically, so what filament does is the only role of filament is to hold the anther pods to the flower. Okay, so what filament will do is it will just hold the anther pods to the flower where the anther can actually stick and not fall off. When the flower reaches the stage of dehiscence, in that case the anthers will burst open and the pollens will be out in the environment. Now, there are various ways by which this pollen can actually get into interaction with ovule and go ahead with the fertilization. So, it can either be via air fertilization or via different animals fertilization. So for example, some plants choose animals for transporting their anthers or pollens to the flower which contains ovary, or there are other ways which there are such plants which the anthers are actually stuck on some kind of structure which is very light. So when the wind comes, the pollen sacs can actually go away in the air, and wherever it gets settled, if it finds the suitable plant containing an ovary, it will just stick to it, and then it can you know so in that case it will just stick to the plant which contains ovary, and it can fertilize it. So the function of style and stigma. Suppose if a plant, so there has to be something which will receive these pollens, right? Ovaries cannot be out present in the environment. It can never happen that ovaries literally like present outside the flower and the pollens will just come and stick on it. That will never happen. So basically stigma is the first base where these pollen grains can actually stick, and basically it's an organ which receives the pollen. Okay, so when these pollens will fly away and it will get stuck in the stigma, it will travel through the style and then it will reach ovary. And in the ovary, it is able to go ahead with the fertilization, which is where the pollens will actually get fertilized with the ovule. Okay, so these are the two reproductive organs of the plant. One is androecium and the other one is gynoecium. So going back to our question, which was which of the following is the male reproductive organ of the flowering plant? The answer is androecium. Okay, so let's go ahead with the next question. Okay, uh, which of the following is the energy currency for a living cell? I'm sorry, I think there was some network issue. What was the last thing that you could hear? Can someone tell me? Am I audible?
Hello, ma'am.
Yeah, yeah. What was the last thing that you could hear?
You just said about you will show the parts of androgen and gynoecium, ma'am.
Okay, okay. So I'll restart it. Okay. So okay, my screen is visible, right?
Yes, ma'am.
Okay. So yeah, we were talking about male and female reproductive organs. So I actually asked a question. I'm not sure if you people heard it. Did you hear the question that I asked after showing the slide?
Yes, ma'am.
So does anybody know the answer to it? That why do plants choose to place male and female
Androecium,
reproductive?
ma'am.
Okay, okay, not that. Okay, I think you missed it. I mean, the network went away quite before. So I'll just start again. So basically, yeah, the answer is correct. It's androecium, and basically, androecium is made of two words. It's andro and ecium. Similarly, gynoecium is also made of two words. It's gyne and ecium. So the word gyne is always associated with the female reproductive organs, and word andro is always associated with the male reproductive organs. Okay, so ecium word basically means the arrangement of these reproductive organs. So when we say androecium, it basically means the arrangement of male reproductive organs, and when we say gynoecium, it basically means arrangement of female reproductive organs. Okay, this I'm not able to see the screen whether there is some issue in the network. So just keep responding so I'll understand that there is no issue. Okay.
Sure, ma'am.
Okay. So, yeah, so let's talk about female reproductive organ first. So, the female reproductive organ basically consists of style, stigma, ovary, and ovules. Ovary is the very, very basic organ for any female reproductive part, which contains the ovule. Now, the ovule is nothing but the single celled female, basically, the cell which contains the female genetics. And similarly, the male genetics comes from the pollens, right? The pollens are basically contained in the pollen sacs, which are contained in the anthers. So, anthers and filaments are nothing but the male reproductive organ, which is called as androecium. Now, the question that I asked was that why do we need this style and stigma? Okay, why is there no provision where the pollens can directly meet the ovule? Or in other words, what I'm trying to ask is that are you aware of that why does there are certain plants which keep their males and female reproductive parts separately? So either they are present on different flowers or they are present on different plants. So why does this happen that the during evolution the plant chose to put these different organs on different locations? Why are they not present together? Does anybody know the answer to this question? Okay, so I'm assuming nobody knows the answer to this question. So basically, the answer is that with evolution, the plant always chooses to, you know, go ahead with reproduction with different kinds of cells just to avoid the inbreeding depression. So the reason that these two organs are present on different parts or different organs is so that the plant can actually get fertilized from the cells which is different from that particular plant. Okay, so now in that case, when anthers will actually get released, they need something which those pollens can settle on. Okay, because the ovaries are not present out in the open. So in that case, what happens is the stigma actually does the work of receiving the pollens and then transfer it through the style to the ovary where the ovule is present and the fertilization it is going to happen. Okay, so that is the role of stigma and style in this female reproductive part. Now, what is the can any one of you tell me what is the role of filament in the male reproductive part? What is the role of filament? Okay, so I'll tell that filament is actually the only role of filament is to hold the anther to the plant so that it doesn't fall off due to any mechanical activity or due to any air activity. That is the role of filament. So, winding up with this, the answer to our previous question was that the male reproductive organ in the flowering plant is called as androecium. Now let's go ahead with the next question. What is the energy currency for a living cell? Can anyone answer this?
ATP, ma'am. Adenosine triphosphate.
ATP, correct. So ATP is the answer to this question. Now let's just understand a little basics about ATP. So yeah, so the full form of ATP is, does anybody want to answer or should I just say it?
Adenosine triphosphate.
Correct. It's adenosine triphosphate. Now, there are, if you know that ATP, there are three things actually. There's ATP, then there is ADP, and then there is AMP. Okay, which stands for adenosine triphosphate, adenosine diphosphate, and adenosine monophosphate. Okay, so according to the words that I'm just speaking, it's quite easy to understand that adenosine triphosphate will contain three phosphate groups, diphosphate will contain two phosphate groups, and monophosphate will contain one phosphate group. Okay, so basically ATP is nothing but a molecule which consists of ribose, which is a kind of sugar, a 5 carbon sugar, and an adenine group attached to that sugar. Okay. Now, the energy of this entire molecule is actually stored in its phosphate group. So, if you see the structure here, I'll just turn on the laser. Yeah. So, if you see this structure, so this is the ribose sugar, which is a 5 carbon sugar, and this is the adenine structure. Okay. Adenine structure contains this NH2 group, and this is the phosphate group. Now, basically, the phosphate groups, they are the bonds by which these phosphate groups are attached together are the ones which actually contain the energy. So, the energy in this ATP molecule is stored in those bonds. So, what happens is when any kind of cell actually requires energy to process any reaction or any mechanism that has to happen in any kind of cell, what it does is it basically removes one phosphate group. And every time a phosphate group is removed from this ATP molecule, some energy is released. Okay, so now if you are aware of the chemistry, some a little chemistry here, the first phosphate group is the easiest to get removed. Okay, so the threshold energy for that phosphate group to get removed is very less. So the first phosphate group removes easily, and the energy that is generated by the removal of first phosphate group will be required to go ahead with the subsequent reactions. But the threshold to remove the second phosphate group is a little higher. So that is why if you see in most of the reactions, ATP is usually converted to ADP. But in certain high energy consuming reactions where it requires much more energy and also it is able to provide it that much of threshold, only in those kind of reactions ATP gets converted into AMP where two phosphate groups are released and twice the energy is also released. Okay, so this is how this basically ATP. This is how ATP actually provides energy to any kind of cell. The reason it is called as currency. So if we talk about the word currency, it is something that you spend to earn to basically get something. Okay, so if a cell wants to get some work done, it will have to spend something. So in this case, the cell has to spend some ATP to get some work done. For example, if I am talking right now and if I'm moving my hands, so there are certain muscle contractions that is happening. Okay, so if my cells want those muscles to contract, basically it has, so that muscle contraction is the work that my cell wants to get done. So in order to get that work done, it has to spend some kind of currency, and that currency is basically ATP. So to move every muscle in our body, the body is basically spending ATPs. Similarly, in case of, for example, plants, if you're talking about plants, plant is known to photosynthesize, right? So every time a plant is photosynthesizing, that is the work that the cell wants to get done, and for that work it will spend ATPs as a currency. So that is the reason why ATP is called as the energy currency of the cell. Now we'll just talk a little about ATP. So basically, ATP, as I told you previously, that ATP basically stores its energy in the bonds which is present in its phosphate group, because that is the only bond that can be broken easily. The bond between adenine and the ribose sugar basically cannot be broken that easily. The threshold for that is very high. So that means the cell will have to spend a lot of energy to reach that point where it is able to separate the adenine and the ribose. So the easiest bond to break is the phosphate. So that is where the energy is stored. Okay. Now how it releases the energy once, so basically that the process of removal of that phosphate group from ATP is called as hydrolysis. So hydrolysis is nothing but it will just release the H2O. The reaction will actually remove the H2O, the water, and that is how the phosphate group will get removed from that. Okay, so this energy release process happens through the hydrolysis of the phosphate group of ATP. Now, as I told you that in case of animals, this things like muscle contraction, like nerve impulses, various synthesizing of various chemicals in the body. In case of plants, photosynthesizing, transportation of certain things, conversation of one substrate to another substrate for subsequent reactions. All of these things are basically the work that needs to be done for the cell and by the cell, and for which the cell will spend its energy currency, that is ATP. Okay, and from this, so basically all the ATP is usually produced in the mitochondria through cellular respiration. It can be either produced by respiration, or it can be produced by beta oxidation, or it can be produced by ketosis. So these are the only reactions where ATP is generated in the cells. If I try to tell this in a very normal local language which is easy to understand, when we eat food, so there are certain kind of foods which get broken down uh, via these processes, the respiration process, the oxidation process. So these processes are nothing but they are just generating ATPs for our body, and these ATPs get stored and they will be utilized for every work that the cell needs to do. Okay, so that's how the energy is being produced in the cell, and then it is stored for the later function. Now, basically, there is one more thing that ATPs can do is it can also transfer the energy to make another reaction happen. So basically, whatever energy is, so suppose there's a reaction where one compound has to be converted to another compound, and it requires some threshold of energy. So in that case, what will happen is ATP will remove its phosphate, and from the energy that is being generated from removing this phosphate group will be utilized to convert one molecule to the other molecule. Okay, so basically this is some basic information that you need to know about the ATP, and which confirms basically the question to our answer. Sorry, the answer to our question, that is ATP is the energy currency for any living cell, be it plants, be it animals. In all the living organisms, ATP is the energy currency. All right. Now let's move ahead to next question. Okay. So cell found around the stomata which regulates the pore size is called as.
Sorry to disturb you, ma'am. We are not able to see the PPT.
Uh, okay. I'll just.
Right now it is visible. Thank you.
Okay, so it's not visible in the presentation mode. Is it visible now?
Yeah.
Okay. Is it
Yes, ma'am.
changed? Are the slides changing?
Not yet.
Has it changed now?
Is a third question we are able to see right now.
Or what are you able to see?
Third question.
Okay, okay. Yeah, yeah. We are on the third question. So anybody knows the answer to this question? Okay. Is it okay now?
God salesman?
Yes, correct. So the okay, I think now the screen should be changing properly, even in the presentation mode, right?
Yes,
Okay.
yeah.
Yeah. Okay. So the question, okay. So the question was that cells that are found around the stomata which regulates the pore size. Stomata. Okay, yeah. Whoever answered this question, great. The answer is guard cells. So now, what is stomata? I think it's a very basic question to ask at this point. I'm sure all of you have studied this. So stomata is nothing but basically the small pores which are present on the leaf of the plant from which the plant is basically able to respire. Okay. So now this is how a stomata looks like, right? Um, yeah. So this is what a stomatal cell. Has anybody ever seen stomata under a microscope in their practical classes or anywhere like that?
No, man.
Okay. Um, okay, in next session I have images of the stomata, but I just don't have it right now. So I'll show you how the stomata looks under microscope. So it's very beautiful actually. If you go for this surface electron microscopy, the images of the stomata are actually very beautiful, and it's very evident that certain stomatas are at the same time, some stomatas will be wide open, and some stomatas will be like opened really small. Some will be completely closed. I really want to show you that, but it's not possible. So okay, maybe next class I'll show you. So yeah, so this is how stomata, like the diagrammatic representation. So if you see the cells under microscope also, it will look exactly like this. Okay. So just assume that there are two kidney beans, the rajma that we eat. So you just put two kidney beans together, and the hole that is created in the middle of that. So basically that is a stomatal pore, which you can see here. This pore is called a stomatal pore. So now how the stomatal pore is created? These two kidney beans like structure here, this and this, these are basically the guard cells. Now these guard cells are everywhere surrounded by the epidermal cells, which is the normal cells of the leaf, and these guard cells will have its own chloroplast. It will have its own nucleus as well. Now how this actually works, let's just understand that. So mm, let's take some other example to understand that how a guard cell will work. If you take two balloons, okay, two round shaped balloons, and if you basically remove the air from those balloons, the two balloons will come close to each other, right? So there won't be any space between them. So it is the original shape of that balloon is something like this. But when you don't fill air in that, it will become flaccid and it will come together, which will actually close the pore that was previously been formed because of this curved structure, right? But when you fill the air back again, the cells, the basically the balloon that we have will take its original form, and it will again form the curve, and that curve will basically give the space to form a little pore in that. Okay? That is exactly how the stomata works. Now just instead of air, in this case, it will take in the water content. Because of that water content, the basically the stomatas will swell, and that swelling will cause that pore to open. Okay, so every stomata that is present in the leaf is every stomata. It's not like there is some huge bunch of stomata which is surrounded by guard cells. No, basically the guard cells are the one which form the stomatal pore. Okay, so that means every stomata is surrounded by a pair of guard cells. There are always two. That is why it will form a small hole in that. And the guard cells, they have the ability to change their shape. They can change their volume in response to the environmental signals. So now, why is it said that they will respond to environmental signals? What do we mean by environmental signals? What is meant by environmental signals? Can someone answer this? Anyone wants to answer that? How can guard cells basically,
Sunlight,
yeah,
like sunlight, air.
Sunlight, yeah, sunlight, correct. Sunlight is like the reason stomatas basically work. Yeah. So if there is more sunlight, so the stomatas, they'll need to do photosynthesis, and for that the stomatas will open. And otherwise, at night, stomatas usually get closed. Yeah, one reason is sunlight. Is there any other? Okay, pH. Somebody answered. Okay, Komal Verma answered pH, temperature, light. Correct. So the light is not the only reason. The plant, the stomatal cells, the guard cells basically can also respond to change in pH of the cell content as well, of the environmental content. The temperature can vary. The stress, the different kind of stress. There's more rainfall, less rainfall, more humidity. So for example, if we take the example of, say, what do we say, suppose waterlogging. Okay.
Transpiration, no?
Yeah, yeah. What?
Is it transpiration, no?
Transpiration, correct. So transpiration happens because of all these kind of reasons. If a plant is under stress condition, any kind of stress, it can be a drought stress, it can be heat stress, any kind of stress. So that stresses will basically cause the plants to um, you know maybe, so suppose we'll talk about, say, waterlogging stress. So waterlogging stress is nothing but excessive water in soil. So in that case, what will happen is that transpiration is basically stopped because plant is in excess of water. Okay, there is more water than it is required for the plant, and in that case, the plant will just, it will try to evaporate it. So in that case, the stomatas will open more. So what will happen because of that is ki the stomata can respond to different stresses also, even in case of humidity. So when the environment is more humid, that itself causes some kind of stress in plants, which will actually, I'm sorry, there's no electricity, so avoid, I mean, ignore my video lighting. Yeah. So in case of different stress conditions, what will happen is this plant will control its transpiration rate. So not just light, but this can be controlled by different environmental signals as well. Now, now there's this line in this, the paragraph that is written is, it can control the exchange of gases between the plant and the atmosphere. So what happens is, the opening and closing of stomata is not just associated with transpiration, but it is also associated with intake and release of various gases, like oxygen, like carbon dioxide. Some plants are also able to intake various chemical gases. There's one plant called spathiphyllum. I'm sure a lot of you have seen that plant. It's very beautiful, usually used for decorative purposes. So it is said that that plant is able to consume a lot of toxic chemicals that are present in the air. So these things also happen through stomata, for which the guard cells is responsible. Okay, so that is why it is said that it can exchange gases between the plant and the atmosphere. Now, okay. So now let's just talk about some key points about the guard cells. So as we have been talking for very long, that the functions of these guard cells is to regulate the size of stomatal pore by changing its turgor pressure. Turgor pressure is nothing but the turgidity pressure. So there are two states of the cell. One is called as flaccid, one is called as turgid. So when the cell is basically swollen up and it has all the water content it requires in the cell, that state of cell is called as turgid state. And when the water content is removed and basically the cell has less water content and it will not retain its shape, in that case, that state of cell is called as flaccid stage. Okay, so the stomatal pores are regulated by the turgidity pressure, the amount of pressure, the amount, basically the pressure is nothing but the it's a way of saying how turgid the cell is. The more the cell is turgid, the more will be the turgor pressure. Okay. So with this turgor pressure, it will regulate the size of the stomatal pore. And the mechanism, as I have been telling you for very long, the mechanism is nothing but the water intake. The more water that will be intook by the guard cells, the more it will become turgid, the more it will have turgor pressure, the more it will expand away from each other, and more the pore size of the stomata. that will cause more exchange of the gases. And now, when there will be some case where the guard cells will lose its water, in that case, the cells will become flaccid, and that's when the pore will close. Okay, is it clear till now? Whatever I have been telling about guard cells?
Yes, ma'am.
Okay, great. So as I asked the question that other than light, what are the other cues? How does the guard cell understand that this is the time where I should intake the water? This is the time where I should lose the water that I have. So there are various factors. One of them is definitely light. The other one is the carbon dioxide levels, the humidity levels. All these things will indicate the plant that now you should um, close the stomata. You should now open the stomata. Okay. So yes, the answer to the question was the stomata pore size is regulated by guard cells. Okay, so we'll go ahead to next question. What are apical meristems? Are they primary meristems, secondary
Primary
meristems?
meristem.
Primary meristem, correct. So can you tell me what a primary meristem is? Okay, so let's just talk a little about apical meristem. Apical meristem is nothing but the topmost tissues which basically divide. So it is a meristematic tissue. Now, what is the function of meristematic tissue? That it is able to generate new tissues from itself. Okay. The reason it is called as apical is because it is present towards the end. It can be either present towards the shoot end or it can be present towards the root end. So basically, the roots will also have root apical meristem and the shoots will have shoot apical meristem. Okay. Now,
Yes, ma'am.
uh, s- yeah. So apical meristem, basically, how does it work? So what happens is, um, I guess there are some people who are joining. Let's just take this 5 minutes break so these people can join. And I'm still having students who are joining, so they are not able to actually come in because of this presentation. Just give me 5 minutes, okay? We'll restart in 5 minutes. Okay. So yeah, let's just start. Yeah, so we're talking about apical meristems. Yeah, so as I was telling that meristems, so basically what meristems does is, the meristematic cells are the only cells which have the ability to divide. Okay, so these cells will be able to produce new cells around it. Now, if it is present on the root, so basically that is how the plant will be able to go ahead uh, in expanding. Either it can be roots or it can be shoot. Okay, it can expand both roots and shoots, right? The roots of the plants will also grow, the shoot of the plant will also grow. So what will happen in that case is, because there has to be new cells which will produce the plant, the parts of the plant, that those cells are the meristematic cells. Um, does anybody know that which there's one technique where apical meristems are very useful? Does anybody know what is that technique? Why
Tissue
are
culture, ma'am.
tissue culture correct? So um, basically this, since because we're telling that these apical meristems are newly produced cells, what happens is, usually the apical meristematic part of any plant is considered as disease free. Okay, so because since it is newly produced, usually the cells are have, even if the plant has any kind of infection, it will most likely, it would have not reached that part of the plant. So in case of tissue culture, we always require a bunch of such mother cells which are not infected in any way. Okay, because if you take any plant cell which is infected, either the culture will get contaminated, or in other case the plant that is being produced won't be that healthy. So that is the reason where apical meristem is very useful. And so yeah, so the first ability of apical meristem is cell division, then it is tissue differentiation. um okay, let me tell you the third function also. It is organ generation. um why so um I'm sure everyone knows this fact that a group of cells will form tissue and a group of tissue will form organs. Okay, so basically the base of forming an organ is cell only. So if any bunch of cells are able to generate new cells, they will definitely be able to generate new tissues and definitely be able to generate new organs as well. Okay, so when these apical meristems basically divide, and if they reach that stage where it has collected enough cells to produce a tissue, in that case, those particular cells will divide and form a group of very specific tissues. Like either it can form, epidermis of that cell, or it can form some vascular parts or some ground tissues or things like that. So when the after multiple divisions, the cell will combine and form one tissue. Then it will start getting differentiated to form a specific tissue. Now, when these tissues will combine, so when you see a plant growing, if you observe a plant, initially when you plant a sapling, it might have just 3 or 4 leaves. Okay, but when you take that same plant, say after a month or so, there might be 10, 15 leaves. If it's a flowering plant, there will be some 5 7 flowers on it, some fruits on it. If it's a fruiting plant, so that flower, those leaves, those fruits are also a bunch of cells. Okay, so certain cells would have divided to form tissue, and that tissue would have formed that organ, that leaf or that fruit or that flower. So if apical meristem divide enough to form a bunch of tissues which can combine to form an organ, that is when a new organ generation happens. New organs could either be leaves, it can be buds, it can be a different shoot as well. Okay, you see plants which have various branches. So all those branches are nothing but they are they are the formation of organs. They are different organs. The branches, the leaves, everything is a different organ. So these apical meristems can also be used to generate new organs. Okay, so are we clear about this apical meristems? This is the basic information which will help us give the answer. Okay, so the question still remains is, that how is it called as primary cell, primary meristem, and secondary meristem? Okay, so to answer that question, basically primary meristem, it is nothing but those kind of meristems which will generate the first generation cells. Okay, first generation cells are basically the new cells that is being generated in the plant. When any kind of plant will form a new leaf, or it will form a new, say, uh, a bunch of anything that is new, in that case the cells that will work will be primary meristems. But suppose that there is an injury to plant. Okay, suppose there is a herbivore which chewed on some leaves. In that case the plant will also repair itself. Okay, so those meristematic tissues which will help the plant in repairing, that will be basically called as secondary meristems. So the reason apical meristem is called as primary meristem is because it will be helping the plant in formation of new organs, in formation of new tissues. Okay, and that will lead to the growth of the plants. Secondary meristems will not lead to growth of the plant. It will basically help in defense anything that is secondary. Like there are different things. There is something called secondary metabolism as well. There is a primary metabolism, there is a secondary metabolism. So in that case secondary metabolism is nothing but it will be that kind of metabolism which will not be an essential thing to for the survival of the plant, but it will be required for the plant to fight against defense or to fight against adverse conditions. So in that case that will be very important. Okay, so here also when we are talking about primary meristem or secondary meristem, so primary meristem will be any kind of meristem which are generating new cells for the growth of the plant, not for the survival. Secondary meristems will be those cells which will be utilized in the survival of the plants, like defense, like repairing the leaf. That will also require generation of some cells which will help repair the wound or some things like that. So that is called a secondary metabolism. And that's where your answer comes from, that apical meristem is a primary meristem. Okay. Okay. So this is the last question for our today's session. And let's see which one of you can answer. Which of the following is the precursor for auxin hormone? Does anybody know the answer to this? Auxin hormone? Yeah, Monica.
Tryptophan,
What? Sorry?
tryptophan,
Yes, it is tryptophan. Um, okay, so correct. It's tryptophan. The precursor here means, so basically auxin is a form of, so basically all these things that are present, any kind of hormone, uh, all these things are nothing but proteins. Okay, they are a form of proteins, and all the proteins are formed by nothing but amino acids. When we say something is a precursor, it is nothing but in the reaction, that is how, that was the first thing, and then our final product was formed. Okay, so there are multiple pathways which have a precursor and which will lead to the reactions forming certain, any kind of hormone, any kind of these kind of things. So in case of auxin hormone, the precursor is tryptophan. Um, let's talk about what auxin hormone is here. Okay, so tryptophan, as I already answered the question, it is that tryptophan is the amino acid which is being used to produce IAA. So IAA is indole acetic acid, basically. Okay. So indole acetic acid is a chemical structure which forms which is basically the auxin hormone, which has the property of auxin. Now, what can auxin do? Um, Okay, so there are two kinds of growth that a plant can do. It can either grow like in a vertical direction, the apical meristems will keep on dividing and the plant will grow in length. But suppose it's a flowering plant. Okay, so in case of flowering plants, there are certain locations. So, for example, the pea plant or the moong plant, the legumes. Okay, so what will happen in legumes is that legumes, they are only produced from where the branching will happen. that essentially translates to the more the branches, the more the pods it will produce. So if the plant continuously keeps on uh, growing in the apical direction, or only the apical meristem keeps on dividing, the plant will grow in the length and it will not branch out. So there are certain mechanisms in the plant which can you know produce lateral branches. Okay, now suppose when plant realizes that there are too many lateral branches, and I don't need the bushiness in the plant, I need the height. Okay, or I need the tips to expand, for example root tips. Okay, so if the apical meristematic things will keep on working, or suppose apical meristem stops working, in that case there are chances that the roots will not grow. Okay, now this entire mechanism, this regulation of either apical growth or uh, lateral growth, this is regulated in plants, and all these things will happen because of a very tightly regulated hormonal mechanism. One of the hormones is auxin. So what auxin does is it is basically involved in apical dominance. Apical dominance is nothing but when the auxin is produced, basically the apical buds will inhibit the lateral growth. So in that case, what will happen is this plant will keep on growing in the length direction. The height of the plant will increase, and there will be less branches. So if suppose there is any plant which will just produce the flowers at the tip, or which will just produce any kind of fruit at the tip, there are certain auxin treatments which are given to the plant. Okay. Or in case of tissue culture, so if in case of tissue culture we want the roots to grow fast, or we want the shoots to grow fast, in that case the media it is added with the auxin so that these tips can grow. This apical growth can enhance and the plant can increase its height. Okay, so that is the function of auxin. Now auxin basically it can affect various chemical, so it can also be formed by the breakdown of carbohydrates, which are basically called as glycosides. Can anyone tell me, there's IA is one of the examples of auxin. There's one more such chemical which works as auxin. Does anybody know that? There's one more chemical.
IPA, indole
IBA.
butyric acid.
Correct. So IBA is another chemical which is also one of the very important auxin hormonal content. So basically these things, these are the hormones which regulate. There are very many different kinds of um, hormones. So plants are very. Because you are such a varied audience, I am not very sure that how many of you have botany as their subjects. But for those who do, they know that plants are really very interesting, and they have such um, I don't know what to say, such interesting mechanisms to regulate everything, to control every all of these things. The crosstalk between hormones is so tightly regulated that a slight change in the amount of hormone can make the plant go from a very tall plant with single flower to a short plant with multiple flowers. So that much ability, the regulation, that is how tightly these plants regulate their mechanisms by various hormones is one of those processes. There are multiple such processes by which plant regulates it. But yeah, I mean, I'm a botany student, so I'm just talking about plants. But yeah, so all I want to say is that auxin is one of the very important hormones which are involved in the regulation of the apical growth of any plants, and also it will basically inhibit the lateral buds. So if there is more auxin in any plant, the lateral growth of that plant will be inhibited. Clear? Okay. So this was basically the first round of your discussion session. We will have few more rounds like this. We will keep on uh, basically discussing various. So these questions are actually previous sessions questions, and we are discussing that only. This will just help you solve your assignments, because we are I don't have the permission to help you solve those assignments, but I'm sure this is how you will have to solve. You have all the study material. Other than that, if there is any doubt that you want to discuss or you want to ask, we can go ahead with that. Otherwise, we'll just close the session. And Yeah, just let me know if there's anything else.
Ma'am, may I know how will be the exam pattern, ma'am? It's like OMR or like question and answers, ma'am?
Okay, I am actually I'm just a TA, so I won't quite be able to tell you the answer to this, but I'm sure that you have been instructed well. You have the course instructions. You can go ahead. So as I think as far as I know, you already have your assignments, right? You have already received the assignment for first week.
Yes, ma'am.
So those assignments are OMR. This it's not even yeah multiple choice questions, MCQs, right?
Yes, ma'am. Yes, ma'am.
And I think this your final exams will have some centers where you'll have to go and give the exams, if I'm not wrong.
Yes, ma'am.
Yeah. I'm not sure because I'm just a, it's I'm just a TA. I just have to discuss these questions. I won't be able to answer you this question, but I'm sure that NPTEL has very well discussed the instructions of your exams. You can go to their website, go to the SWAYAM website. I'm sure you will find all these things there. I'm not the right person to answer this.
Yes, ma'am. I can understand. Thank you, ma'am.
Yeah, if there's anything else regarding the topic, or I'll be taking the next 3, 4 sessions of whatever. I think there will be 4 more sessions, 3 more sessions. Yeah, this was the first one. We'll have 3 more sessions. So if there's anything else and that you want, if there's any specific topic that you want me to teach you in the next session or anything other than whatever I discussed today, please let me know so that we can include that in the next session as well. Otherwise, the sessions will go on like this. So I'll just give you the previous questions. We'll discuss those questions, go ahead with the topics, and they'll just help you solve the your assignments. I'm not allowed to discuss that. So yeah, if there's anything else that you want me to teach you specifically, please let me know. The next session will be on the same day, same time, next week, Tuesday, 7 p.m. to 9 p.m. So if there's anything else, please let me know. Okay, so should I close this session?
Yeah, sure. Yes,
Okay.
ma'am.
Okay, then have a good evening and I'll see you next week. Same time, same day. Okay?
Thank you. Good evening, ma'am.
Okay, bye.
Thank you, ma'am. Thank you,
Okay.
ma'am.